Welcome back, everybody. My name is Kevin Fryert. I run a small company called Salem Oaks, which I started right after I finished my career at Pfizer after 30 years doing R&D in many different roles. Salem Oaks is setting out to empower patients to shape the future of medicine by educating them, preparing them to talk about drug discovery and development. I also sit on the board of Rare New England, and I'm on the advisory board for the Cure Mito Registry. And I really enjoy this morning's discussions, and it's exciting to see all this work going on. We've heard a lot about the preclinical work, the basic science, learning about the mechanisms, all the different avenues people are looking at for Leigh syndrome. And now we're going to shift gears a little bit into how do we actually discover drugs for that and develop those drugs and then push those drugs through the clinic so that we can understand which are the ones that we should be moving forward. Today, I'm joined by Dr. Ann, who's the Vice President of Biology at Cytokinetics, Dr. Matthew Klein, who's the COO at PTC Therapeutics, and Dr. Volkmar Leissig, who's a professor of pharmacology and biologic sciences at Midwestern University in Glendale, Arizona. So Ann, why don't you get us started?
Okay, you see the full screen?
I do.
Okay, great. Thank you so much to the organizers for inviting me. It's so wonderful to be amongst these very esteemed researchers doing very exciting research that are, making significant progress toward finding treatments for these diseases. My job here, though, is to sort of give you an overview of the drug discovery process. So it is primarily targeted toward the patients and families that are tuning in today, to sort of give you an overview of what this is like and the some of the challenges that we face when we try to develop drugs for mitochondrial disease. So I will there we go. So I don't want to go over all the details of this. What we've talked about a little bit today, what we talked about this morning was a good bit about gene therapy and the approaches that are being taken to bring gene therapy to the clinical sites to treat these patients. As you could hear, this is a fairly long and involved process to make sure that it's not only going to be effective, but also safe. The other topic that we've talked about a little bit in the previous few talks was using what we call small molecule therapy, or the conventional concept of a drug, which is a small molecule. Aspirin was the first drug to be discovered back in the 1890s. And what you can see here is the progression of the modes of therapies that have been developed over a century and how it is progressing. And of course, gene therapy is down here on this list as it's come basically to the drug marketplace only fairly recently in the 1990s, but it holds a lot of promise for patients with mitochondrial disease. So what I'm going to talk about and focus on in this talk is sort of the process of this more conventional form of the discovery of small molecule drug therapies. So here it is sort of depicted the entire process, and that is we start with an idea or a target. For instance, in Dr. Mutha's example that he gave, he did some screening to identify genes that allowed cells to escape a mitochondrial defect. And those genes then told him that one approach to treating this disease could be, for instance, lowering the oxygen tension, which is then going to enhance the ability of the cell to make ATP, which is so critical to organismal function, by the glycolytic path and not be so reliant on mitochondrial ATP production. So that process of identifying a target can be a multi-year process. So the target discovery and followed by some initial compound discovery. What you just heard in the previous talk was taking a cell model system, throwing onto a high throughput plate a whole bunch of compounds and looking for how it changed gene expression. And so that's an aspect of compound discovery, looking for exactly what the effect of the drug is going to be and could it be beneficial in the model that you're most interested in. So all that process takes 2 to 3 years, sometimes longer. Then we move on to non-clinical studies, and this period can take 3 to 5 years. Non-clinical studies means basically moving that compound that you have identified, usually in a cell or even a biochemical assay, and moving it into animals and finding whether the drug is effective or not. And I'll touch on some of the details of that in just a moment. But I'll sort of finish out the process here of then going through clinical studies, which is probably one of the longest of those phases. You just heard 2 talks that are potentially interested in using already FDA-approved drugs and repurposing them for use in these patients. And to do so then means cutting out this entire long period of clinical studies that are required to get to bring a drug to market. Takes about a year after a compound has been through clinical studies, and if it's successful, the FDA is going to make a decision that takes about a year. If you get approval, then you can launch, means take your compound to the drug market, and then it'll start be used in patients. And it's imperative that whoever the company is that developed the drug continue to monitor in those patients that are using the drug for safety and efficacy as time goes on. So, but what I want to do here is then explain, because this whole process of the laboratory and animal early kind of discovery studies can be broken down into additional steps. I talked about target identification, validation, and you got to develop an assay, some of which were described. The assay in the last talk was looking at how compounds change gene expression, and that was the assay format. And you've got, that needs to be high throughput. So he was describing they do those in 384 well plates. They've made major leaps and bounds in the huge amounts of data that are generated from that kind of experiment to pull through that, to get to condensed information that's going to tell you whether your compound was effective in the direction that you wanted it to be. So when you find a compound, say it's not an FDA approved drug, then you're going to keep iterating through a process of taking that drug into lead optimization. And lead optimization means you've got to understand how the body metabolizes that drug. Our livers and other organs in our body will take a drug that you've ingested and they will change it. And it can be changed to be better, or sometimes can be changed to be toxic. And so we want to make sure that you can modify the molecule in a way that's going to get around that metabolism. You've got to modify that molecule in order to optimize the half-life in the bloodstream. Some drugs, reach the bloodstream or only high in the bloodstream for an hour or 2, and that might be enough to be beneficial for the patient. For some other indications, you need a level of drug in the bloodstream to persist for many hours in order to have efficacy. There are all sorts of other aspects, solubility. You've got to be able to get a drug into solution when it's in the bloodstream. If it falls out of solution and precipitates, it's going to cause major problems. And so there are all sorts of chemical characteristics about a small molecule that have to be optimized. And so the medicinal chemists take the data that's generated in all these assays, they look at the compound, they make some additional compounds, tweaking the structure of the compound and the physicochemical properties of the compound. And then you reiterate back through the process, back through the screening assay, and back through animal studies. And those animal studies, as you've already heard today, are very often mice, sometimes rats. And then from that point, you'll do toxicity studies, which are also done in small molecules, but the Food and Drug Administration requires you to do toxicity studies in larger animals as well, once you have a lead compound that you have confidence in. And so at that point, then you can file an application with the FDA for what's called an investigational new drug, or an IND application. Once you have that IND application approved by the FDA, you can move to clinical studies. And here are the different phases of clinical studies that I think many of you have already heard about, and I thought I'd detail some of the aspects of these studies that are inherent to this process. In all of these studies, one of the primary metric is how safe is this compound. If it's never been in human beings before, the utmost importance before it goes on to be tested in a large population is that it's safe. So a phase 1 study is exactly that. It's a safety study, but you're also aiming to figure out what dose is going to be appropriate. You're going to try to figure out whether it's tolerated. Does it very often drugs will create either some nausea or a headache. Sometimes even though that's not life-threatening or health-threatening, it certainly makes patients uncomfortable, and they may not tolerate it and drop out of the study. These phase 1 studies are usually quite small. It's more likely to be 20 to 30 patients big. This 100 would be on the high end, and usually it is healthy volunteers. Not always, but sometimes it is, or most of the time it is healthy volunteers. It takes about several months. And about 70%, because of all those studies you've already done in small and larger animals, 70% of those show that they're safe and can move on to phase 2. And a phase 2 trial is then testing for efficacy. This is where you begin testing it in the diseased patients that you're intending to treat. And again, you're looking for safety, and then you're now also looking for, does it help the disease symptoms? You're also sometimes optimizing the dose. It may involve more than one dose in a phase 2 study, looking for side effects. And you've gone from 20 or 30 patients now to hundreds of patients. This may take months to years, and part of this has to do with the rate of progression of a disease. So for instance, in Alzheimer's disease, the rate of progression and loss of cognitive function is very, very slow. So in order to conduct a successful trial, that trial has to go on for many years because you can't detect a tiny change in cognitive function. The drop in cognitive function has to be big enough in the general population of the patients to be able to see a statistically significant difference as a result of drug administration. About only 33% of drugs move from phase 2 to phase 3. Then in phase 3, these are the big studies. They can take 300 to 3,000. My company ran a heart failure phase 3 trial in the past that was over 8,000 patients. And the reason to have so many patients is because you want your statistical power to detect an effect of the drug to be very high. And the more patients, the higher the chance that you're going to be able to detect an effect. Again, you're monitoring for safety. These studies can be long, again, based on the rate of progression of the disease. And only about 50% of those that make it to phase 3 go on to the FDA for approval. If you have a successful phase 3 trial, then the filing that you apply for with the FDA is fairly highly likely to be approved. So that new drug application that takes approximately a year to prepare and a year for the FDA to review, then is fairly likely to be accepted. And that's when you can then be taking your drug into the marketplace. But I need to point out, and this is part of the challenges in drug discovery, is that starting from the point of clinical studies, the likelihood of success to getting your drug to patients is only about 10%. There is a huge fallout due to safety, due to off-target effects that you didn't anticipate. I'm sorry, but mice and rats are not humans. And so you will begin sometimes to see toxic effects in human beings that did not show up in smaller species. And so this sort of summarizes some of the challenges in basic drug discovery. But I think that there are, yes.
And
yeah,
we're seeing a little reminder screen down the corner of your screen.
Oh,
Can you
sorry.
turn that off? It's covering up some of your slides.
Yes.
Thank you.
Sorry, I can't. Put my cursor to move in the right direction. Sorry.
I guess don't worry about it, because
No, I know. We had this
your
problem
cursor's not
earlier
helping you.
and I can't get it to disappear because I can't get the pointer. Excuse me, just a moment. There we go. Okay, so how does this relate to then mitochondrial disease? So what I wanted to point out is that in a subset of diseases, no, it won't go. In a subset of diseases, for mitochondrial disease and neurological disease, there are basically only two drugs that are approved for treatment of the disease. And one is, both are actually ROS reactive oxygen species scavengers. The rest of these that are listed for metabolic disease and cardiovascular disease are also FDA approved drugs, but they may or may not relate to what we need to use to treat mitochondrial disease, but they do modify metabolism in ways that could be considered for repurposing. But only some of these would be used for that purpose. So why are there so few diseases, so few drugs approved for these diseases? The total number of FDA approved drugs is about 20,000. There are approximately 50 new drug approvals per year. And like I said before, approximately 10% of those that enter clinical trials actually become approved drugs. So why is that? Why is it so challenging? Well, I think we've heard some of those challenges in the talks that we've gone over this morning. And that is, there is a huge complexity in mitochondrial disease, in that there are two genomes involved in creating functional mitochondria. And so one of those is, and this may be particularly for gene therapy for mitochondrial DNA encoded mutations, that is the biggest complexity that has kept us from moving forward. But as Dr. Minjig detailed this morning, major progress is being made there. For nuclear encoded mutations, gene therapy is actually significantly easier. And there, as was detailed earlier, there are some success stories related to other monogenic diseases where there are FDA approved gene therapies. And that will, we all anticipate, will continue to grow for the nuclear encoded mitochondrial diseases. Other challenges include the ability to design and execute robust clinical trials. This is one of the reasons why the registry that is open to these patients is so important for all the families to participate in, because what that does is allows those companies and the NIH, et cetera, that want to run a clinical trial to know where those patients are and to know what their clinical presentation is so that they can identify the patient population that could enter a clinical trial. Numbers are an issue for those. Obviously, there are ATP6 mutations and SIRF1 mutations that are more prevalent than others. For those that are quite rare, a clinical trial may be quite challenging in that if it's a gene modification trial, it's going to be very specific for that mutation. And so the number of patients that are available to conduct that study is critical to the success of the trial. We've also talked about, Dr. Mutha and others have brought up the topic of biomarkers. We can't sample the brains of these patients, obviously. So you would prefer to have either a urine or a blood-borne marker of improved function. And I think that as time goes on, we're going to come up through efforts like what Dr. Mutha was describing and doing proteomic profiling of blood samples, metabolomic profiling of blood samples to identify biomarkers, ready biomarkers. As we heard, the MRIs are great for diagnosis. They would be difficult, in particular for young children, as a monitor of disease status. Another big component that has, I think, hampered development of drugs for these diseases is risk aversion. And the good news is we see, particularly in the last couple of years, we see significant progress in overcoming the fear of the risk of failure. Big pharma companies, for instance, like Astellas, have acquired mitochondrial-centric companies like MitoBridge. So there is an appreciation in big pharmaceutical companies that obviously have huge resources, that going after mitochondrial defects is worth the investment. And that also has been enabled by changes at the FDA for how trials can be designed for rare diseases and what is acceptable to the FDA for the patient sizes that need to be tested in order to get an approved drug. So things are changing both on the drug development side as well as at the FDA to enable this risk aversion to maybe be lessened. Limited expertise in mitochondrial function was always a problem years ago in pharma and biotech. Most mitochondriologists in those companies were relegated to being toxicologists, and that is just looking for the off-target or toxic effects of compounds that are often at mitochondria. But again, this is changing, and it seems to be changing quite rapidly. As all of you, I think, know from what has been presented today, the complexity of metabolism pathways and the complexity of mitochondria is huge. Sometimes when you add a drug to cells or an animal modeling the disease, you don't have an effect. And if you study it long enough, you may find it's because the organism has compensated for the presence of the drug. That is, our metabolic pathways are very complex, and you can tweak one pathway just to have the cell upregulate another pathway that sort of makes it as though you never added the drug at all. There are quite a few proteins that make up mitochondria for which we don't have structures of the protein, and protein structure is actually very important for drug discovery so that our medicinal chemists can look at the structure of the protein and fit a molecule into the three-dimensional structure of the protein, like at its active site or at an allosteric site. But even more so, there are some activities in mitochondria that we know are very important. Some of these are at the mitochondrial membrane that we still don't know what genes encode those proteins. And so we know there's a function, but we don't know the genes that encode it. When you don't know the gene, it makes it quite difficult to study in drug development. Conservation of target across species was touched on earlier as well. We run a program where a compound that we've developed works a bit differently in mice than it does in higher species. And it's because the sequence of our target is different in mice than it is in human species. And so that can be a challenge to the development of drugs for these purposes. And all right, I'm going to, in the effort to cut down on time, I'm going to skip some of these next slides. This is simply an example of where we didn't know what the calcium uniporter was made of as far as the proteins that did it when we were hunting for calcium uniporter inhibitors. And as a result, we were completely unsuccessful at finding drugs just based on a functional screen. But others have had more recent success in finding mitochondrial uniporter, calcium uniporter inhibitors, because actually Dr. Mutha was one of the ones to discover the components of the calcium uniporter. And because of that, we now know the structures of those proteins and can then more easily target molecules to them. I think I'll skip over these next so we can get to some summaries. One thing I'll say about this, and with regard to metabolism, we have been working in the area of NAD boosting, and actually boosting of NAD with niacin has been shown in a publication by a Helsinki researcher, mitochondrial disease researcher, Anna Suomalainen, who saw that NAD boosting with niacin, which is a vitamin, could help these patients. And this is one of those examples where the complexity of the pathway makes it a bit more challenging. If we want to discover something that's a bit more robust than niacin in producing the end product of the pathway, which is NAD, you can see that this metabolic pathway is really quite complex. And so that can hamper the discovery process. This is another example. When you go to modify the amount of NAD in a tissue by boosting with nicotinamide or nicotinamide riboside, look at the differences that you can boost in different tissues. In the brain, the ability to boost it is fairly low. In the kidney, it's dramatically higher. And we don't have a complete understanding of why that should be. And so these are lacks, there's a lack of knowledge about this that hampers our discovery process. So to summarize, given the great news is that given the unmet medical need in mitochondrial disease, the drug discovery efforts will continue to expand, and you've heard great examples of how that is taking place in the drug discovery world right now. The willingness to accept those risks is now decreasing. The annotation of the mitochondrial proteome, which Dr. Mutha's group and others have gone a long way to help us achieve, has improved. And we're now getting, that has not been really touched on, tissue-specific drug targeting is gaining momentum. So if we need to get a drug specifically to the brain or specifically to the heart, there are ways to contemplate doing so. And obviously, one of the biggest hopes for all of those suffering from these diseases is gene therapies. I think Vamsi made the comment, though, as well earlier, that gene therapies are probably going to still need to be combined with small molecule therapy, as most gene therapies have not been completely restorative of function. And so maybe you can tweak the treatment a bit by adding a small molecule that raises the success rate of the therapy. So I'll close now.
Thank you.